Then I am going to start the new subject that is going to called as the bioprocess engineering and the process biotechnology. See here. So the next is the bioprocess engineering and process biotechnology. So here first I am going to start the production process. So exactly how we are going to produce the antibiotics. So first I am going to explain about the production of an antibiotic. So how the antibiotics are going to produced here. So the first is the production of antibiotics, and in the production of antibiotics I am going to explain about the mainly the penicillin, the tetracycline and the aminoglycosides antibiotic, and the aminoglycosides antibiotic. So the first that is going to called as the bioprocessing engineering and the process biotechnology. So the first topic I am going to start, that is the production of antibiotics. So how the antibiotics are going to produced here. So first antibiotics I am going to start, that is the penicillin. Then the second antibiotic which is going to called as the tetracycline, and the third antibiotics that is going to called as the aminoglycosides. So how the antibiotics are going to produced here. So first I am going to explain the production of an penicillin antibiotic. See here. So how exactly the penicillin antibiotics is going to produced. See exactly about the penicillin antibiotics. So the first exactly, what are the antibiotics here? So if you can see the antibiotics, so the first point of the antibiotics is that antibiotics are the chemical substance it is a chemical substance that can kill microorganisms or inhibit their growth. Exactly. And see here, they are the products of secondary metabolism, है ना? And exactly, है ना? So there are two different types of antibiotics which is going to used here. So the first antibiotic that is going to called as the broad spectrum antibiotics, and the broad spectrum antibiotics are that they can control the growth of several unrelated microorganisms. So they are going to control the growth of several unrelated microorganisms. So see here exactly, the example is the tetracycline, and the second example that is going to called as chloramphenicol. Then the next that is going to called as the narrow spectrum antibiotics, The narrow spectrum antibiotics, so they are effective against selected species of bacteria. They are effective against selected species of bacteria. Selected species of bacteria, and exactly here is the penicillin and streptomycin. Penicillin and streptomycin. So exactly see here, so a large number antibiotics, so they are going to produced by the microorganism. So the microorganism are going to produce antibiotics. Microorganism are going to produce antibiotics and they are the products of secondary metabolism. And they are the products of secondary metabolism. They are the products of secondary metabolism here. So the first point of the antibiotics is that, see here, so what are the antibiotics? So the first point is the antibiotic production. So what are the antibiotics? Mainly it is a chemical substance that is going to kill the microorganism, or they are going to inhibit their growth. Exactly. So the first point of the antibiotic is that is a broad spectrum antibiotics. So the broad spectrum antibiotics means they are going to control the several unrelated microorganisms, and it is going to consist of an tetracycline, and it is going to consist of an chloramphenicol. Then the second point, that is going to called as the narrow spectrum antibiotics. So the narrow spectrum antibiotics means they are going to effective against the selected species of the bacteria, and it is going to consist of penicillin and streptomycins. And mainly these antibiotics, so the microorganism is going to capable to produce antibiotics, so therefore they are going to the products of secondary metabolism. So they are going to the products of the secondary metabolism. So first I am going to explain about what are the different antibiotics and how they are going to produced by the different microorganisms in the process biotechnology. See here. So first I am going to explain about the production of an penicillin. So how the penicillin it is going to produced. So the first antibiotics which is going to produce, that is going to called as an penicillin. So how the penicillin is going to produced, I am going to explain here. See exactly about the penicillin. So the first antibiotic production, so that I am going to explain is the penicillin. So how the penicillin it is going to produced by the certain types of, so how the penicillin it is going to produced here. So first antibiotic production I am going to explain about, that is the penicillin production. So the first antibiotic, penicillin. So see here about the penicillin. So the penicillin are the group of beta-lactam. So they are called the beta-lactam bacteria antibiotics. So they are going to consist of the beta-lactam ring, and exactly they are going to kill the bacteria. Therefore they are going to called as bactericidal ring. And exactly the first antibiotics it is going to discover, the first antibiotic it is discovered. And exactly if you see the structure of the penicillin here, so the basic structure of the penicillin, so it contains a lactam ring, it contains a lactam ring and thiazolidine ring, and thiazolidine ring fused together to form 6-amino penicillinic acid. 6-amino penicillinic acid, exactly. So see here, so the first point is that about the penicillin. So the penicillin that is going to called as the beta-lactam bactericidal antibiotics, exactly. And this is the first antibiotics. This is the first antibiotics that is going to be discovered here. Then after it is going to contains a lactam ring. And what is exactly the lactam ring? And the second most important point, it is going to consist of the thiazolidine ring. So the thiazolidine ring and the lactam ring, when they are going to fuse together, to form 6-amino penicillinic acid, then exactly what is the structure of the penicillin? See here, so this is the sulfur, and here you can see double bond O, there is nitrogen, there is hydrogen, there is NH, and exactly it is going to links with the C double bond O R here, and here is the CH3, that is going to called as C double O H, and here is the H. So here this is going to called as the lactam ring. This is going to called as the lactam ring, and exactly this is going to called as, this ring is going to called as thiazolidine ring. So when we are going to combine the lactam with the thiazolidine ring, that is going to called as 6-amino penicillinic acid. And this R group may be different in the, and this R group may be different in penicillin G, ampicillin, amoxicillin, So exactly, see here, so this is the structure of an penicillin. So the penicillin is going to consist of the two different ring. So the first ring that is going to called as the lactam ring, and the second ring that is going to called as the thiazolidine ring, and they are going to form the 6-amino penicillinic acid, and the R group may be different in the penicillin G, ampicillin and the amoxicillin. Exactly. So the first point of the penicillin is that it is going to consist of an beta-lactam bactericidal antibiotics. See here. Then after I am going to explain about the action of the penicillin. So how the penicillin is going to highly effective against the gram-positive bacteria. Then the next is the action of penicillin. See here. Then the action of penicillin. Then see exactly the action of penicillin. The action of penicillin. So the natural penicillin are highly effective against penicillin are highly effective against gram-positive bacteria. The first point, they are going to highly effective against the gram-positive bacteria. They inhibit the cell wall synthesis. They are going to inhibit the cell wall synthesis. So how the cell wall synthesis is going to inhibit? Because they are going to block transpeptidation reaction. They are going to block transpeptidation. So they are going to block the transpeptidation reaction, and exactly many microorganisms such as S. aureus, Staphylococcus aureus produce beta-lactamase that degrade beta-lactam ring and make penicillin non-functional, and and therefore that bacteria is resistant to penicillin. So that bacteria is resistant to penicillin. And exactly, see here, so the action of penicillin. So the penicillin are going to be highly effective against, the gram-positive bacteria. Then the next point is that they are going to inhibit the cell wall synthesis. So the cell wall synthesis is going to completely inhibited by the penicillin, and they are going to block the transpeptidation reaction. So the transpeptidation production reaction is completely going to inhibited by the penicillin antibiotic, but there are certain microorganisms, है ना, exactly, there are certain microorganisms such as Staphylococcus aureus. So Staphylococcus aureus, है ना, they are going to produce beta lactamase. So exactly, if they are going to produce the beta lactamase, so they are going to degrade the beta lactam ring, and if the beta lactam ring is going to destroy here, the penicillin is going to become non-functional and the bacteria that is going to become resistant to the penicillin. So exactly, this is the action of the penicillin. Then after I am going to explain what are the microorganisms which is going to used for the production of penicillin, then the microorganism used in the penicillin production. Then microorganism used in penicillin production. See here, so what are the microorganisms used in the production of penicillin? Organism for penicillin production. Organism for penicillin production. See here, the Penicillium notatum, Penicillium notatum was used for the large scale production of antibiotics. For the large scale production of penicillin, but nowadays the Penicillium chrysogenum that is going to used here. So the nowadays the penicillin, the chrysogenum, see here, but the nowadays the penicillin chrysogenum that is going to call as the Penicillium chrysogenum, and its improved mutant strain mutant strains are preferred, and its improved mutant strains are going to preferred here. Exactly. So this is the two main important topic, है ना. So this is the two main important, the microorganism which is going to used for the production of the penicillin. Exactly. So how the penicillin it is going to produced here. So the first point is that the Penicillium notatum. So the Penicillium notatum is going to used for the large scale production of penicillin. For the large scale production of penicillin, the notatum is going to require, as well as the Penicillium chrysogenum. So the chrysogenum, its improved strain or the mutant strain, nowadays it is going to preferred for the production of penicillin. Exactly. Then after I am going to explain what are the biosynthesis process that is going to associated with the penicillin production. So how we can going to produce penicillin. So what are the chemical reactions which is going to occurs inside the microorganism for the production of penicillin. See, So exactly what are the reactions here?